All right, so hello and welcome back. We're going to start in on antibody-mediated immunity. And I really like this picture here. You're noticing in here on the left, we've got this virus. It has like all these different kind of surface proteins, slightly different shapes. There's some red ones, purple, kind of round ones. Um, and you'll notice there's different antibodies binding to them. And then you'll see these antigens here on the right. Again, they have these different surface protein markers, and again, very specific antibodies binding to them. Those surface markers, and let's see if I can get my pen to work. So these surface markers, they're calling them determinants in this image, but we have another name for those. We call them epitopes. Epitopes, E-P-I-T-O-P-E-S. So on the surface of each one of these viruses, not unlike any of our cells, there's many different proteins on the cell surface. So each one basically, let me kind of back up. So here we have this virus, right? If this gets gobbled up by like an antigen presenting cell, those different MHC2 molecules on the surface of the antigen presenting cell may present slightly different epitopes. So perhaps one will present the circular one, another one will present this sort of red triangle one, another antigen presenting cell will present the purple one. Okay, so they'll present different ones. And so different, really T cells and B cells will respond to different ones, these different epitopes. Okay, and in this case, what you're looking at in this image is that different B cells have responded to the different epitopes and have produced slightly different antibodies in response. Okay, so when we're talking about antibody-mediated immunity, what we're talking about is antigens being destroyed by antibodies, antigens being destroyed by antibodies. This works particularly well against antigens in the bodily fluids, extracellular, meaning things that are outside of the cells, extracellular pathogens, things like bacteria, things that multiply in bodily fluids but do not enter body cells. Once they're inside cells, again, the antibodies cannot help you. That's the job of the T cells. Okay. And again, really, most of the time, pathogens provoke both types of immune responses. It's not just the B cells attacking. It's not just the T cells attacking. It's both parts of your immune system is going to attack the foreign invader. All right. So let's talk a little about the antigens and antibodies some more. Okay. So those antigens are little bits of, or molecules or bits of foreign material. It could be the entire microbe, as you're seeing in this part of the image here. It can be parts of the microbe. It can be bacterial toxins, pollen, again, transplanted organs or incompatible blood cells, like we saw that. Again, the transplanted organ part is only going to refer to the T cells attacking that, okay, but it still is an antigen. We talked about antigens, again, in the red blood cells, and we talked about the A antigens for type A, B, Rh for B positive, those, okay? So things that you have, well, I guess certain characteristics you have to be considered an antigen. So one, it has to be able to produce or provoke an immune response. Okay, so it's not considered an antigen if it does not provoke an immune response. Okay, that word antigen is actually a contraction of two words. So antigen is actually a contraction of antibody generating. So antigen. So basically, it has to provoke that immune response, and it has to be reactive. So it has to have the ability to react with cells or antibodies it caused to be formed. Okay, so if you have, I don't know, this virus cause antibodies to be formed, but those antibodies can't interact with it, it's not considered an antigen. And I really can't give you guys a good example of when that would occur, so I apologize for that. I can tell you things that do not provoke immune responses or things like, I don't know, bits of metal in you. And that's why they're able to like put plates inside of you to hold bones together, or plastic. Things like that would not provoke an immune response. Okay. Again, we have self-antigens that identify our own cells. We've already discussed that. So here we have, again, is the antigen. Here are the epitopes that I was referring to in that other image. So again, as this antigen gets gobbled up by an antigen-presenting cell, that antigen-presenting cell may present slightly different epitopes. Okay. And we, so again, your immune system can recognize and respond to a billion different epitopes, which is an amazing amount, really. But it also explains how, you're able. You get a little bit sick, but you don't die of colds and flu viruses that change every year, right? Unless again you're immune compromised, maybe elderly or sick with something else. So basically, each B and T cell, as I was mentioning to you, are going to be specific for a specific epitope. Okay, so that all this is trying to say is that it has a very unique segment that will code for a unique antigen receptor. So like this B cell maybe responds to this epitope. Another one will respond to this one. Another one responds to this one. Maybe another one will only respond to that one. It just depends. Okay. All right. So antibodies are proteins that can combine specifically with the antigenic determinant or that epitope on the antigen that triggered its production. Okay, so that's all it's saying. So this will bind to whatever it was that caused it to be produced in the first place. Taking a closer look at an antibody, they're actually glycoproteins. Again, that means they're part sugar, part protein, and you can see that here, part of that carbohydrate chain is what gives it that glyco part. There's a special term for them. They're called immunoglobulins. Most contain 4 polypeptide chains. You see like the heavy chains as well as some of the light chains. Too heavy, too light. There's what's known as a hinged midsection. So right in here, you can kind of see that little wavy kind of squiggle, which basically means that this part right here can move. So it's a little bit flexible. So think of like going from like a Y, like your hands up in the air, like as a Y, to like a T, arm straight out to your side. So it can kind of flap a little bit. And that makes sense because remember the antibodies are going to bind with something at the very ends of those chains. So here you can see it's the antigen binding site. So it makes sense that they can then kind of move around a little bit. It gives a bit more flexibility. Okay, so I just said. All right, so we have 5 different classes of antibodies. We have IgGs. These are the most abundant, and we'll see what they look like in a second. They help protect against bacteria, viruses. This is the only class to cross the placenta. So guess what? The antibodies against Rh would be in the IgG category. IgA can be found in sweat, tears, saliva, and you can kind of see this in all these different secretions, which means they provide a localized protection. All that means is IgA, if it's found in sweat, well, guess what? It protects you on the surface of your skin. If it's found in tears, it protects your eyeballs. It's not going to protect anywhere else other than the eyes. Saliva, mouth, etc., etc. Okay. IgM, you can see it's the very first, it's a very quick to respond one. It's the first antibody to be secreted by plasma cells after initial exposure to an antigen. And this is also one of the ones that causes agglutination to occur and the lysis of microbes. Right? IgDs, not very plentiful. These actually act more of receptors on the surface of B cells. They're kind of antibodies embedded into the surface of a B cell. And you have IgEs, and these are involved in allergic and hypersensitive reactions and help protect against parasitic worms. So this is just what all those sort of look like. Okay. You're not going to get an image on a lecture exam and go, is this, and try to identify it, so don't worry about that. Okay, so how do they actually work? Well, I think we had a little bit of insight from the way they would work with red blood cells, right? They could cause agglutination, so they kind of clump things together. But they also help neutralize, so they block or neutralize bacterial toxins and prevent the attachment of viruses to body cells. So let's just kind of think about that for a second. The way that a lot of toxins are going to work is that they're going to be able to bind to a receptor or somehow get across that cell membrane by having a whole bunch of antibodies attached to the toxin or to the virus. It prevents it from actually entering into the body cell. Again, if they're attaching to the bacteria that have those little tails, the flagella or cilia, by attaching to them, it prevents the movement of the cilia or the flagella, so the bacteria can't move. Well, if it can't move, it's not going to be very hard for that monocyte or that macrophage to come and gobble it up. Okay. Agglutination, as in the red blood cells. There's what's known as an activating complement. So basically, it can act as as a sort of a signal, which is then going to basically acts as a signal, which basically causes those phagocytes, the monocytes and such, to be particularly attracted to the microbe, and they will gobble it up. And you can see they'll also enhance blood vessel permeability, again, attracting those phagocytic cells. Right? And it may even help create channels in the plasma membrane, so kind of like poking holes in the plasma membrane. Not unlike perforin, kind of very similar in the end result. And again, as I mentioned, it helps enhance phagocytosis. It acts as a signal to those phagocytes. Plus, when you kind of clump a whole bunch of things together, that also will attract those macrophages and the monocytes to the area to gobble things up. Now, we've used antibodies in science. We've taken advantage of their ability to attach to these epitopes. So what you're looking at here is a cell, and you can see there's in this case, just A and B antigens, those two different epitopes on the surface of the cell. And what you see, this little purple thing, that is an antibody. And what we've done is we've artificially manufactured it. So you add a fluorescent tag to it. So on every single antibody, there's a little piece that fluoresces. So if you shine the right light on something, it will glow underneath the microscope. So, for example, certain cells will have very specific markers on their cell membrane. Like, we know this through trial and error. So let's say you're interested in seeing if a body region has, I don't know, let's do something very simple, like a cancer cell. So cancer cells will have very specific antigens on their surface, right? Even epitopes. So what you can do is you can create this antibody with this fluorescent tag, take a section of tissue and go, let's see if there's any cells here that are showing us those cancer markers, okay? And if they are, the antibody will attach to it and put in the microscope, it'll glow, okay? You can also do this in things like, in this case, what you're looking at is a section of brain, and everything in red here is producing something called corticotropin releasing hormone. It's part of your fight or flight response, part of that your stress cascade. And any cell that's glowing red happens to have that hormone. This is actually part of my graduate work. And as a little bit of an aside, the way you do this, you can see you have this goat anti-rabbit and rabbit anti-A. So let's say if we're looking at a human cell, what you try to do is you want to create an antibody that's going to be very artificial. So something that it is going to be very specific to that A. Like if I made this in another human, it might have cross-reactivity with other antigens in the human body. So in that case, what I want is something that's very different than us, like a rabbit, and I will artificially make this so that it attaches to the A. So it's an anti-A antibody. And then I will produce another one in a totally different animal that will only attach to things that are rabbit. Does that make sense? So there should be nothing else rabbit in you or in those cells that you're looking at other than the thing that you added. So basically what this allows is for magnification of the signal. So it doesn't just glow, it grows really bright. Hopefully that makes sense. Anyway, what we call this is immuno, because we're using principles from the immune system, antibodies, histo, because it's histology, and trust me, there's lots of chemistry also involved. So scientists just smushed all three words together, and they called this technique immunohistochemistry. Oh, and the other nice thing that you can do is you can then pick different colors for different proteins. So you can go like, well, let's see, what cells are producing like corticotropin releasing hormone at the same time that they're producing epinephrine, for example. And you can actually look at that to see if they're close to one another, are they not close to one another, what have you. And that's what you're looking at right here, actually. So again, that's corticotropin releasing hormone and actually norepinephrine in these cells. And it's gotten way more sophisticated than that over the years as well. And again, that was some of my work in case you're interested. All right. Not only do we have lots of different T cells, but we have millions of different B cells. Again, each capable of responding to a very specific antigen. But unlike the T cells that go out, the B cells stay put in those lymph nodes or lymphatic nodules or the secondary lymphatic organs and allow the antigens to be brought to them. Okay. Once activated, they will differentiate into plasma cells, and it's that active form that secretes the antibodies. So that's what we're looking at here. Here's an inactive B cell. A microbe comes along. It can activate it. Remember, B cells are also antigen-presenting cells, so they don't necessarily have to have the co-stimulation by the helper Ts, that interleukin-2. However, when that helper T offers the co-stimulation, the response is much more robust. Okay, it's amplified. So basically, the same thing occurs as we saw in the T cells once these B cells are activated, in that they're going to divide. They're going to divide and they're going to differentiate. Okay, so you get many of them of the active form and then also the memory form. So basically, that's just what I just said. Okay. And so they'll become B cells. B cells will turn into plasma cells as well as memory cells, again, the reserve force. They only secrete one type of antibody, so either they're going to secrete the IgA, IgM, whatever the type is. But oh my goodness, look at this. A plasma cell secretes hundreds of millions of antibodies each day for 4 to 5 days. Hundreds of millions of antibodies every day, just one little plasma cell. That is immense. That is incredible. Sometimes it just does not cease to amaze me, the numbers that our body deals with sometimes, right? So those antibodies, because obviously the B cells are in lymphatic tissue, so they'll travel through the lymph and then into the blood to the site of infection. Okay. So we have what's known as immunological memory. So what you're looking at here are two different types of classes of antibodies. So in red is the IgM, in blue is the IgG. So what you're seeing here is, here we have our first exposure to whatever the microbe is. And then, so on the y-axis, we have antibody titer. So basically, how much antibody do we have in the body? And on the x-axis, we have just days. So here we have 0 exposure. And then, about a day or so later, what we start seeing is an increase in these IgM numbers. So we start seeing this response by the increased production of these antibodies. And then it takes a little bit longer for those IgGs to get going, but then they also come in. And then what you're seeing here after this 14-day mark is that the IgM class actually will go back kind of to baseline, but the IgGs stay elevated because of some of the cells already primed. Okay, and so there's a lot more of this IgG kind of floating around. Now, if you get exposed a second time, what you end up seeing is that those plasma cells and the memory B cells that produce that IgG, they remember. So they're much more efficient and much more, robust in their response to that second infection. So those IgG numbers are way higher. Again, this means that you might get that cold a second time, but you never even realize it because they're so good at responding to it the second time. Okay. And those memory cells, something to remember, they can remain for decades, for almost your entire life, right? Right, and also, and the same thing's going to be true for cytotoxic T cells. So this is not just the plasma cells that this is the case for, but it's also the cytotoxic T cells will also show this much large enhanced response. Okay. And this is just all what I just said to you before. Okay. We talked a little bit about cytokines. We talked about interleukin-2. That's really important as the co-stimulator. We talked in a previous video about interleukin-1, which causes fevers. But again, as a general category, these are small protein hormones or chemical messengers. They're not. I don't want you guys to think that they're hormones like in like the endocrine system level of hormones. Okay, but they're definitely chemical messengers that stimulate or inhibit many normal cell functions. So things that they will do is they'll increase growth, cell differentiation. So basically, they're going to stimulate that as well as cell division. Some other examples where there's perforins, the granzymes, we've already seen that, and again, that interleukin-1. Okay. Oh, that should say lymphocyte. It's a typo. So it's not lymphocyte, it's a lymphocyte. The M is missing. So those T cells and some B cells will produce the cytokines. APCs, those are antigen-presenting cells. Fibroblasts, endothelial cells, monocytes, hepatocytes, kidney cells, lots of different cells secrete cytokines. We have different ways to acquire immunity. Naturally acquired active immunity is basically when you get sick and then you go through the illness and then you recover. That's naturally acquired active immunity. We have artificially acquired active immunity. This is what happens when you get a vaccine. Okay, so it's artificial because you wouldn't have gotten that. Sorry, that was my ice maker. You would not have gotten that naturally that way, right? That you inject it into the person, this weakened form of the virus or just a part of the virus, and then your immune system mounts a response. Artificially acquired passive immunity, sometimes for very medical reasons, they'll simply inject the antibody itself. And so instead of waiting for your body to produce the antibody, they'll just inject you with it. Now, the downside of this is that your body does not then start producing it itself. Okay, the nice thing with these first two, with the naturally acquired active and the artificially acquired active, is that once you're exposed, you start producing it yourself, and there's a memory for it. With the artificially acquired passive, there is no memory for that. Basically, once those antibodies, get broken down in your body, you're not producing anymore. And there's naturally acquired passive. So basically, this is when antibodies are passed from mother to fetus. All right. I'm actually going to stop that right here because this has already gotten pretty long. So last video, we're going to talk about what can go wrong in the immune system. All right. See you guys next time.